Okay, good afternoon. Let's see. For those of you who are taking this course for credit, sometime next week we will have the final exam posted. This is a multiple choice electronic exam. You needn't lose any sleep over it, and you can take it as many times as you want. If you complete the stated number of questions, you'll get a certificate for having participated in at least 50% of the sessions. That's the sort of a hallmark for coming. Now, next week is the last sort of double lecture-like series, which is on the mitochondrion and mitochondrial diseases. So that will be posted electronically in some detail a little bit later. Lynn Wolf from the Clinical Center and Jennifer Lippincott-Schwartz from NICHD. And then in the very last session is this mini symposium on what does the future hold for PhD scientists, those who are interested in translational work and those who aren't. And so we're very fortunate. There are 4 institute directors and John Gallen, who's the head of the Clinical Center, who will briefly discuss what, in their view, the current situation holds and what opportunities there are and what excitement there is in their field. And the whole thing will be a general discussion, questions, and it's usually, this is an annual event, which is not only well attended, but there's a great deal of interaction. Okay, so today, Jeff, thanks. Okay, so it's a little over a year ago we had a session on Parkinson's disease, and I received a fair number of emails suggesting that we have another one. And part of the reasoning was because from a clinical standpoint, the disease itself was fascinating enough and challenging for many people who didn't know much about it. And from the standpoint of research and science that's going on, there's really like an explosion of different modalities that are being brought to bear. And previously, we had an extraordinary presentation of sort of neurosurgical approach to control of tremors and influencing rigidity. And so today we'll hear about another exciting avenue that comes to bear to diseases like Parkinson's disease, and that's really a sophisticated cell-based neuroscience, and in this case using a technique called optogenics, which you'll hear about. So Parkinson described this shaking palsy disease, but in his description and in much of the literature, this is pretty overt, and you're going to see some of it on some movies in a moment. But I would emphasize that this, well, that makes it mutual. I don't know who you are. Oh, very good. I'm Win Arias. I'm the course director. Okay. All right. So the point I wish to make is that this, like many neurological diseases, is very insidious in the onset. And some of the people in whom it's most apparent, this insidious onset, are people who do finely tuned things like being a violinist or being an athlete. The first indication that Lou Gehrig, the famous baseball player, had what later be called Lou Gehrig's disease was when his batting average began to progressively decline, although he continued to play ball. I had a tennis partner who used to beat me regularly and mercilessly, and then I noticed that he wasn't quite as quick with his step and as forceful with his serve and everything else, and I looked at him much more closely and realized that, my gosh, he had early manifestations of Parkinson's disease. So this is kind of an interesting parameter to think about the earliest phase of a disease rather than the overt one. And I like to make mention in this context of a man whose name has sort of been lost a little bit in history, but to me is one of the medical giants. He happened to be a friend. George Kotzius was a physician scientist at Brookhaven National Laboratory who basically asked himself, why is the basal ganglia in the brain, the substantia nigra part of it, why is it nigra? Why is it black, whereas much of the other brain of the area is white? And so he isolated from that area the chemical dopamine. And George did an experiment which today would be impossible. It would take 20 years before it was ever approved. And he reached up on a shelf and took some D-L-Dopa from a chemical vial. They brought some patients into Brookhaven who had severe Parkinson's disease, and he sort of titrated them and showed for the first time that their tremors disappeared. And this was really the basis for the pharmacological treatment of Parkinson's. So he was somebody who discovered something and did something about it in an era when it was possible to. Now it's still possible, but it's infinitely more difficult. So today we're going to talk about Parkinson's in the genomic, the neuroscience, and the optogenetic era, which I'm sure will be very exciting. So we have two speakers, and I'll briefly introduce them to you. Actually, Kodran Lungu spoke to us a little over a year ago, and he's been kind enough to go to bat for Mark Hallett, who is his branch chief, who was going to speak today, but has been floored by an intestinal virus. So Colin is trained as a neurologist, a psychiatrist, and has a very distinguished record of basic and translational research and clinical trials and so forth relating to tremor diseases, particularly Parkinson's. He's chief of the National Institutes of Health Parkinson Clinic, assistant clinical director of NINDS, and director of the Botulinum Toxic Center, which maybe some of you may wish to ask him about afterwards and gain some information. And our second speaker is a new arrival at NIH, Alexxai Kravitz, who obtained his PhD in neuroscience at the University of Pennsylvania in 2009 and did a postdoc then at the Gladstone Institute at UC San Francisco and came here in 2012 to join NIDDK. Alexxai is interested in neural circuitry at the cellular level, and his work came to my attention when I saw, and I think it was the catalyst, the creation of an optogenetic interest group here at the NIH. And knowing next to nothing about optogenetics, I began to read about it and was utterly fascinated, and particularly by reading some of his work. And so I thought it would be a very exciting avenue for all of us to learn. So thank you both very, very much, and maybe Kodran, you want to begin.
Thank you, Wayne. Can everyone hear me okay? No? How about now? No?
Move it higher.
It's good? Okay. All right. Just a little bit of a lag there. All right, thank you for the introduction. As you heard, I'm a last-minute substitution. I'm sorry, I know how disappointing it is to come and see the star and it's the understudy. But I was told that because of this, the next lecture will be free. So I gave a lecture about a year and a half ago, and I thought it was a great experience. Mark was planning to do this one, so unfortunately, we didn't have much of a notice to change. So this afternoon between patients, I tried to frantically turn this into a merge of his slides and mine. I'll ask for your patience a little bit as we go through this, but I think it will still be useful information and hopefully a nice setup for the second half of this lecture. So I realized just now that there's no disclosure slide. I have some research agreements, but no relevant financial disclosures. Still hoping, though. So as Wynn pointed out, this was first described by James Parkinson in 1817. It's a fascinating read, the original description. Incredible amount of insight, and you can still read it now and still discover, wow, he even knew about this. He may not have understood everything about how the disease comes about, but the description is very interesting. The pathologic hallmark is the Levy body, which is a cytoplasmic inclusion that occurs in specific vulnerable neuronal populations. By the way, it's Levy. He was German, not Louis. I learned that recently. And the gross anatomy pathologic hallmark is the degeneration of these pigmented neurons in the substantia nigra. This is neuromelanin that is present in these neurons. On the left here, you see, on the right, you see a normal midbrain with this dark band in the substantia nigra, which is the main concentration of dopaminergic cells in the brain. On the left, you see the brain of a Parkinson patient with pretty much nothing left there. The major features of Parkinson's disease are bradykinesia, rigidity, tremor, and postural instability. And then there is an entire score of non-motor features. And it's interesting that you mentioned in the introduction that there are insidious features versus the overt ones. There are actually some that for a while are invisible, although the disease is going on. So I'll touch a little bit on that. And in addition to these motor features that have been known for a long time and are the most visible ones, really the non-motor features, as you will see later on, can play a major part and can be some of the most disabling ones. We like to talk about several subtypes of Parkinson's. One way to separate it is into two major subtypes clinically, which is a mostly rigid and akinetic type in which rigidity and bradykinesia predominate. And statistically, this tends to be a little bit more severe and more rapidly progressive, and then a tremor dominant, which tends to be slightly milder. There is a concept now that really maybe considering Parkinson as one disease is simply wrong, and it might in fact be a constellation of conditions that look similar and have related pathogenic causes, but really can be very different. Show a couple of videos of classic tapes. I have to apologize for that band. I didn't realize that I needed some sort of software update here, but you can hopefully still see the classic resting tremor, in this case affecting the jaw, upper extremities, and the legs at the characteristic 3 to 5 hertz rate. It is a resting tremor. It tends to alleviate with action and posture, although there is a action and postural tremor component as well. I think these videos go on for quite a bit, so we'll want to skip with some of them if you don't mind. That was a very interesting phenomenon. You saw how that tremor stopped when the patient moved his arms, and then if you wait a few seconds, it comes back to the same severity. So it's the characteristically resting tremor versus an action tremor. There are other types of tremor that are more prominent with action and posture. Essential tremor is a good example. Akinesia, I don't like this term. It's still there. It technically means no movement. Bradykinesia is the preferred term, which means slow movement or reduced movement. And there's a sequence effect. So characteristically, there is difficulty in performing a fine movement, but also there is a decrement. The movement starts at a certain speed and amplitude, and it slowly gets, you see the amplitude diminishes, and then she tries to override that, and then again there's this diminishing feature. And then it translates into the ability to ambulate and the ability to get up from a chair. This is another feature that we see, and I think she eventually makes it, but it takes her many, many attempts. The masked facies is not listed under the major ones, but it's a very important feature, and I like to point this out. It's actually been shown that this affects the interaction of the patients with their environment, and in fact it affects their interaction with their physicians. And sometimes it's thought that this can impact the quality of care they get, because as a physician, you're dealing with a patient who's just not reacting, he's just not responding to the treatment, and that's been shown to, not at a conscious level, really impact the relationship that the patients have even with their physician. The loss of postural reflexes is also one of the characteristic features, although it typically shows up later, and it can be fairly dramatic. You see at a minor displacement, instead the patient has some difficulty. So there's several steps coming back to catch his balance, and that can be very severe, and in fact the patient could sometimes just fall if the examiner is not there, just go straight down. So you can see that this is a major quality of life impairment and really dangerous, because patients start having falls and other complications. This is a fairly classic Parkinsonian gait with no arm swing. You notice that the arm swing is reduced. There's turning on block. The patient has a lot of difficulty turning. The stride is diminished and the gait is shuffled. And I think we also may have an example of freezing in this video. As you can see, these are very old teaching tapes. You can tell that they come from VHS. This is an illustration of the point I was making earlier, that we might be dealing with Parkinson's diseases. So first off, even if we call it, if we refer just to this classic idiopathic sporadic PD, even within this entity, there are several subtypes. Then we like to sometimes separate out the so-called genetic PD. We now know that we can think of pretty much every disease other than maybe a tooth infection, although even there you can debate as having a genetic component. But there are subtypes of Parkinson's that have a classic Mendelian transmission. They may have major differences in their pathogenesis from the idiopathic Parkinson's. There is secondary Parkinsonism. The MPTP is a famous case, because this also led to the development of one of the best-known models of Parkinson's, the basal ganglia injury. This was a contaminant in a drug of abuse, and those people developed a severe form of Parkinsonism. It can be vascular, secondary to multiple strokes, and then there are conditions that can have Parkinsonism and these features as part of their presentation, but they're really not Parkinson's, and these include multiple system atrophy, progressive supranuclear palsy, corticobasal degeneration, and some of the spinocerebellar degenerations, SCAs, can have prominent Parkinsonisms. This is a list of these so-called monogenic, so Mendelian transmission Parkinson's forms. There are a lot of them, and a lot of them are being discovered, and as you see, for some of them we don't really know exactly what the gene is or what it does, or even the transmission in some of them. I isolated further some of the best known or most common ones. The first one described was the alpha-synuclein mutation. This is a protein that is presumed to be involved in synaptic vesicle trafficking. It's young onset, rapid progression with a high rate of dementia. Really looks very different from idiopathic Parkinson's. The pathology is the same as the idiopathic Parkinson's, though Levy bodies with some tau pathology as well. LRRK2 is autosomal dominant with variable penetration. This is a kinase involved in signaling, and really it's very similar in presentation with idiopathic Parkinson's disease, and the pathology is also variable, so that's interesting. The last three here are all autosomal dominant, and we think that here there is mitochondrial pathology that is primarily responsible for this disease coming about. They have, so Parkin and PINK1 tend to have, actually all three of them, slower progression, primarily motor complication, and the pathology is interesting because it seems to be isolated to the nigra. And I will talk a bit more about what happens in pathology in idiopathic Parkinson's disease, and really nigra is not only not the whole story, but really is just one of many, many areas of the brain that are involved. And another interesting thing is that you actually do not find Levy bodies in these conditions. So really we think there might be something radically different in the way these come about. We do not have a slide with the actual diagnostic criteria. They're not necessarily relevant, but I will just say that the diagnosis remains a clinical diagnosis. And in fact, the accepted clinical diagnostic criteria only go up to probable Parkinson's disease, and definite Parkinson's disease requires pathologic confirmation. And obviously that's an inherent limitation for making diagnosis in these patients. There is a lot of interest in imaging, in developing imaging that can help there. It's one of my main areas of interest in research. Either fluorodopa PET scanning or SPECT or PET scanning looking at the dopamine transporter. They are not diagnostic. They can help. They can help clarify between different types of diseases, whether it's Parkinson's or something else. They really are still at an ancillary test stage. None of them are diagnostic. This is an example of what fluorodopa looks like. So in a control, you see a very strong signal in the striata bilaterally. This is a patient with Parkinson's, and this is a pre-symptomatic stage, and you can see that the pathology is there long before the patient becomes symptomatic. In addition to the motor manifestations, there are a lot of non-motor features. And in fact, many of these are present long before the motor features. And it is fairly clear now that Parkinson's probably begins in the peripheral nervous system, long before it happens in the brain. There is some very interesting research coming out in terms of finding a marker of pre-symptomatic disease. And alpha-synuclein staining on colonic biopsy samples from patients in the VA system who are just getting routine colonoscopies. These biopsy samples looked at 10 or 20 years later when the patient developed Parkinson's. 100% of the people who had Parkinson's were having positive alpha-synuclein staining. So that's very interesting. We're talking decades before the manifestation. So that's work in progress, and we're all very interested to see where this is going to lead. This is not an exhaustive list, and these can be very disabling manifestations. And going a bit more into detail here, this is at 5 years duration. Pretty much 100% of patients have a lot of non-motor manifestations. And in fact, I strongly believe, and I'm not alone in this, that if you don't find non-motor manifestations, by the time somebody has motor manifestations, you just didn't look hard enough, because they are going to be there. And things like fatigue, anxiety, pain, we now know that pain is part of Parkinson's, and sometimes it responds to dopaminergic medication. And a number of other things. Olfactory dysfunction is now considered almost a specific manifestation for idiopathic Parkinson's, so much so that if we see somebody who looks like they have Parkinson's but their sense of smell is normal, we question the diagnosis. This is an interesting study looking at what happens in Parkinson's with a long disease duration. So we're looking at about 15 to 20 years of disease. Really, most of the complaints at this stage are non-motor. End-of-dose failure and dyskinesia were present in all the patients. Falls, dementia, freezing, dysarthria. So you see a number of very disabling manifestations. And since that time when the role of dopamine was discovered, we've become quite good at controlling manifestations of the disease with dopamine. And we've developed a lot of very interesting drugs treating the motor manifestations. We're still really lagging behind when it comes to the non-motor manifestations. So we'll talk a bit about the basal ganglia, and so we put this in context of where these structures are. I'll point out the major players here in a coronal section. This is the substantia nigra. Right above it in this pale staining is the subthalamic nucleus. And then the pallidum and the putamen are here, and the striatum consists of the putamen and the caudate. You see the head here, and the tail somewhere, I think it's labeled on this end. It's sort of that looping structure hit there. So that's what it looks like anatomically. Schematically, this is a simplified view of what this looks like. This is, again, it's a simplified model, and all models are flawed. So keep that in mind. I will take you through the circuits here just very briefly. We like to refer to a direct and an indirect pathway in Parkinson's. So the direct pathway goes from the putamen to the GPI and then to the thalamus. The indirect pathway has a couple of relay stations through the GP and the subthalamic nucleus. A simple way to look at this is to just skip to the end, where we have thalamic nuclei that are considered to provide some sort of a motor tone to the cerebral cortex. So these are stimulating, the gray arrows mean stimulation, the black ones mean inhibition. So this is a stimulating tone facilitating movement. Here's your substantia nigra that sends dopamine to the putamen, and it starts this circuit. If we go over to what happens in Parkinson's, you don't have enough dopamine here. You will see that dopamine has two different actions in the putamen. There is an excitatory pathway and an inhibitory pathway. The net result, and you can, in your time, do the nice math of inhibition of excitation, inhibition of inhibition resulting excitation, and so on and so forth. The net result through both of these pathways is an increased inhibitory output from the pallidum that inhibits this tonus from the thalamus to the cortex, resulting in a diminution of movement. So this explains to some extent the rigidity and the bradykinesia. It does not explain the tremor. And this picture here sort of connects the anatomic view. So this is the path from the nigra to the striatum. It does not explain the tremor. There's a lot of interest in how tremor comes about because it really doesn't quite fit with this picture. This is from a very elegant study in Annals in 2011. The current thinking is that there is a separate cerebello-thalamocortical circuit that is responsible for generation of tremor. And it is connected with this basal ganglia circuit between the GPI and the VOP-VIM region of the thalamus. And the diseased basal ganglia circuit drives the cerebello-thalamic circuit into tremor, causing the Parkinsonian tremor. Talking about molecular pathways here, this gets a little bit more complicated. Again, we'll start at the end where the final result is cell death. You will see that mitochondrial dysfunction features prominently here, and we think that that is central in the pathogenesis in Parkinson's. So the autosomal recessive mutations that I had pointed out, DJ1, PINK1, and Parkin, they cause directly mitochondrial dysfunction, which in turn results in oxidative stress that feeds additional pathways. Looking separately at Parkin, this is a sort of that role exploded. It acts through an intermediary here, and this molecule, PGC1 alpha, has been referred to as the master regulator of mitochondrial survival. So this is just an outline of how that happens. The other side of this equation, alpha-synuclein aggregation is thought to be central in cell death, and also in turn drives additional mitochondrial dysfunction, creating a pathologic feed-forward loop. It's just a very broad outline, and it's very relevant when we talk about gene therapy and possible gene therapy targets. And I'll get to that just briefly later on. In terms of staging of the disease, and this is a topic that I like to emphasize because it really reflects that whole idea of manifestations that you see and some that are insidious. So the first two stages of Parkinson's, this is a pathologic post-mortem study looking at patients with Parkinson's who have died in different stages. The first couple of stages do not even reach the substantia nigra that causes the motor manifestations. They reach the dorsal nucleus of the vagus and locus ceruleus. Then only in stages 3 and 4 is the nigra involved, and this is when you start having motor manifestations. And then in the later stages, the entire brain is involved, including the cortex, causing the dementia and other problems. So what does this do on a clinical stage? Well, there is a pre-symptomatic phase. This is not to scale in terms of time, right? So there's a pre-symptomatic stage, as you can see. I'm sorry, I lost my pointer here. I'm trying to get it. There we go. By the time we have motor manifestations, we think that about 80% of the nigral neurons have already died. So that's a sobering reality when we talk about gene therapy rescuing these neurons. We really need to figure out when this disease starts way before it actually manifests. This is usually when the patients are prescribed medication. There is a honeymoon period when the medication works superbly, and then there's a motor complication period. Then the symptoms tend to become resistant, and then we have a cognitive decline and a period of complications. A brief overview of the treatments. Unfortunately, I'm afraid that that picture doesn't quite show up. So I will just point out, levodopa is the precursor of dopamine. So this would be a dopaminergic neuron. So administering levodopa in combination with carbidopa, which is a dopa decarboxylase inhibitor, this simply provides the missing dopamine. We miss dopamine, we give a precursor, and we essentially replace it. The major side effects are dyskinesia and some peripheral effects, including nausea. Dopamine agonists act down here, so they're not dopamine, but they play it on TV, shall we say. They have the same thing, the same effect at the receptors. They have a different set of side effects because they act on the same receptors, but their function seems to be a little bit different. They have a higher incidence of psychiatric side effects, a higher incidence of drowsiness and sleep problems. Anticholinergics, they really don't fit in this picture. They act on the cholinergic system, so not on the dopaminergic system. They can be effective on tremor. Very little use these days because they have fairly unpleasant side effects. The MAO inhibitors are an interesting class. They do not provide dopamine from outside, but they prevent the breakdown of dopamine inside the central nervous system. And they have a lot of interest because some of them are studied for disease-modifying effect. Amantadine has an unclear mechanism of action, helps dyskinesia. We think it has some glutamatergic inhibitory effects. Again, kind of unpleasant cognitive side effects. L-DOPA remains the mainstay of symptomatic treatment and the most powerful medication. Its side effects are dyskinesias, on-off fluctuations that happen at some point in treatment, and some psychiatric disturbances. This is an interesting study looking at what happens after you start dopaminergic medication. This is dyskinesia, this is motor fluctuation. So after 5 years, almost half, about 1/3 to a half of the patients start having dyskinesia, and it's getting there, about a quarter of patients start having motor fluctuations, meaning they go from on, dopamine is working, to off, and at peak they start having dyskinesia. How are we doing on time? I know we were delayed a bit, but we want to 5-10 minutes, keep going? All right, we'll keep going. This is a slide looking at non-dopaminergic symptomatic therapy, which is an area of great interest. A2A receptors, which are represented here, are an area of great interest. They are abundant in the basal ganglia, and they're concentrated on the medium spiny neurons, which are the neurons on which the dopaminergic cells from the nigra project. And A2A antagonism facilitates intrastriatal GABA release, so there's a lot of interest there. In addition to that, there are drugs being studied that act on the serotoninergic system and others. So we won't go into a lot of detail for the sake of time. Deep brain stimulation has been around for a while, and it's an interesting and, when applied correctly, very satisfactory method of treatment. So simply put, it acts as a pacemaker for the brain. So it does not interfere with dopamine, but it interferes with the consequences of Parkinson's. So it normalizes abnormal electrical signals in the brain. This is a schematic of what the lead looks like. They're placed stereotactically. Can have an entirely different lecture on that. And let me go back to the schematic that I showed you. So there are a couple of obvious targets. If ultimately the result of dopamine not really working is that you have a hyperactive subthalamic nucleus or a hyperactive secondarily globus pallidus driving this whole downstream pathway into inhibition, if you interfere with them here, then you might help the symptoms. And these are indeed the main targets of deep brain stimulation. I don't have time to go into details of how deep brain stimulation works. The short answer to that is we don't know, but we know it works. The longer answer is that at the very least we know it's not direct inhibition. It's not the same as just knocking out these structures. It's a lot more complex than that, and we can talk more about this if we have time in questions and answers. This, since I showed you the schematic for tremor, this would obviously be an obvious target for interfering with tremor, right? It can improve the on time without dyskinesia. So that's when somebody has fluctuations, that's what we target. it generally doesn't do better than medication. So the goal here is to do as good as medication can, but without the fluctuations and the side effects. And a lot of data is coming out now showing that the earlier you do the stimulation, the better you might be. Doesn't mean go to that first, but the early stim trial is the latest thing showing that the earlier you do it, the better it is. Going a bit more into the whole idea that it's not just the dopaminergic neurons. So the gait and falling and the freezing portion that I talked about, these seem to be cholinergic mesencephalic neurons that are mostly responsible for that. And based on those studies, this is an interesting study looking at imaging gait. So essentially it's virtual gait, and the same areas light up in functional MRI as they would in actual gait. And this highlighted the importance of this pedunculopontine area of the midbrain. And this is pathologic data. This is a control patient with Parkinson's disease who did not have prominent falling and gait impairment. This is a patient with Parkinson's who did have prominent gait impairment. So you can see it illustrates the point that all Parkinson's is not the same. So there is a special subcategory of patients that has more difficulty walking. So this led to the whole interest into DBS targeting the pedunculopontine area in Parkinson's disease. And we have a study that we hope we're going to start soon looking at primarily at the physiology, but also at the efficacy. So heading towards the end, I just want to mention the viral vector gene therapy idea. This is a very interesting area that holds a lot of hope, we think, because essentially it allows us to deliver genetic constructs surgically to, for the first time, go to the source of the problem rather than just treat it symptomatically. So you create these contracts where you have a helper or packaging construct that is viral genes from an inactivated virus that then delivers this gene into the host cell, and then the host cell essentially is turned into a medication factory producing the gene product. There are a number of relevant pathways that you can target here. So I'll take it, this is a different schematic of the same thing. So on this area, you can see how you can deliver symptomatic therapy in a much more efficient way. For example, AADC is the enzyme that turns levodopa into dopamine. This is a target using the AAV2, an inactivated adeno-associated virus 2 target that delivers this gene. So essentially you take, this still deals with the neurons, dopaminergic neurons that are still there, and you turn them into accelerated factories of dopamine, essentially. Another clever way to approach this, so these are all studies that are actually either ongoing or in different stages, is, for example, going into the subthalamic nucleus. That was the stimulating pathway, remember, in the schematic I showed you. And if you deliver this enzyme, GAD, you can turn glutamate, which is an excitatory neurotransmitter, into GABA, which is an inhibitory neurotransmitter. So essentially you put a dial in there to dial down the excessive excitatory output. The most interesting one is this. Neurotorin is featured here. GDNF is in the same pathway, and I'll segue into that, which are neurotrophic factors that are supposed to rescue neurons or promote survival. And this would, in fact, go to the source of the disease. So instead of just treating the symptoms, it might actually reverse the main pathogenic problem. So GDNF is one of the several known neurotrophic factors. It was isolated based on a discovery that it promotes survival of dopaminergic neurons. So then logically the next step was to see if it has neuroprotective or neurorestorative therapy. Neurotorin shares a pathway, and you may have heard about both of these. They're both out there in trials. There are a number of factors on this pathway. These only act peripherally. These are the two that act in the central nervous system. And downstream, they promote neuronal survival, migration, synaptogenesis, and maintenance of neuronal populations. The preclinical data looked very encouraging. This is data from primate studies. This is an aged primate model, unilateral injection. You see the successful expression here. And what you see further down, this is staining with neuromelanin, which marks those surviving neurons. So I'm sorry, this is, I'm getting my slides confused here. Essentially, this shows expression of dopamine after the injection of the viral vector. So the aged primate model is probably as close as we can get to a Parkinson's model, because it's not a static injury, it's a progressive degeneration, just like we have in humans. And based on that, there have been a couple of trials. The latest one to come out, I think Serogene just announced the preliminary results of their trial, and it did not meet efficacy criteria, but it met some secondary endpoints, and it was shown to be safe. We have a study that we have just started at the NIH using GDNF, and we've just enrolled our first patient. The first surgery has yet to take place. This will be a dose-finding study, safety and efficacy as a secondary outcome, so we're pretty excited about that. And this brings me to the end, where the take-home points would be that we have some classic features in Parkinson's, but then a number of non-motor features that can be just as or more disabling than the motor ones. We're doing a lot of work in understanding better the etiology and how this disease comes about. We have a number of medications, and for many years we can do very well with the symptomatic therapy. Deep brain stimulation definitely has a major role in increasing quality of life and extending the window of efficient therapy. And I would say the future is bright, so hopefully I'm not giving the same lecture 10 years from now and we're in the exact same place, but I don't think we will be. Thank you. So I think we have time for questions.
I have the impression that sometime in the Middle Ages there are juvenile forms, or could you speak to the role of inflammation?
Yes, that's actually two different questions. So idiopathic Parkinson's disease probably peaks in the 7th and 8th decade, in the 60s and 70s. The pathogenic process starts a lot earlier than that, as we now know. Juvenile Parkinsonism is a different type, and it is most often associated with one of those known genetic causes, sometimes with unknown genetic causes. And in fact, Camilo Toro, who is the neurologist in the Undiagnosed Disease Program, has been kind enough to involve me in some of their Parkinsonian patients. So we've seen some very interesting early Parkinson's patients, and in some of them we did not know how it comes about. The inflammation question is definitely an interesting topic, one that's generated a lot of interest in terms of developing therapies or understanding the pathogenesis. We know that oxidative stress and inflammation plays a role. We do not have a direct linear connection from inflammation to Parkinson's. We don't think that it's a primary inflammatory disease that then causes Parkinson's. So inflammation probably plays a role. We're still in the process of defining what that role is. Yes? Just yell.
With the gene therapy, is there a concern that you need to turn the gene product off at some point, or if you turned it on too strongly, can you go backwards?
Right, that's an excellent question. So the question is, you put a gene construct in there, what happens? Because that will just keep going, indeed. For something like GDNF, if the target that we have is to promote survival of the remaining neurons, logically we say, yes, let it go forever. I don't want to turn it off. For one of the other, say, symptomatic focused therapies where you start expressing a product, it may have an adverse effect that you really have no way of turning it off. So the constructs that have been used in clinic do not have an off switch, do not have like a tetracycline or something like that. So it has been an issue in some of the initial trials where they had like intractable dyskinesias. So it's a reasonable concern. For the neuroprotective gene therapy approaches, the preliminary data, the primate data, and even the preliminary human safety data indicates that it's not likely to be a concern.
I was just wondering if the early Parkinson's has the same symptoms?
It depends on what type of early Parkinson's. So you can get earlier Parkinson's manifestation in LRRK2 carriers, for example, and it can look just the same as the older patients. For the most part, no. For the most part, it goes back to the concept of it's probably not just one disease, but several that kind of look the same. So definitely the one that starts very early in life tends to look different. Generally the pattern, so I'm going to leave aside here the juvenile Parkinson's, the one that starts in childhood, because that tends to be very different. The one that happens, say, in the 20s and 30s, and for example, the PINK1, DJ1, Parkin, mitochondrial-linked so-called Parkinsonisms, they tend to be different. They tend to be primarily severe motor manifestations without the non-motor manifestations. So, yeah, the easy answer is yes.
I seem to recall that Parkinson's is typified by these extrapyramidal cells, triangle-like cells, which also Alzheimer's has and PICS. Is that true? And if it is, what does this type of cell tell us about these diseases?
The extrapyramidal term actually refers to the entire circuitry. I don't think that there's a cell it's an old term that we don't necessarily like a lot, and the idea was that there's the pyramidal cortical neurons that are the corticospinal pathways involved in movement, and then there was a global umbrella term applied to all the other motor pathways that were extrapyramidal. So, that's the pathway that's involved in Parkinson's. But there isn't a extrapyramidal cell type of population.
So is the Lewy body the cause of or the result of?
Excellent question. So yes, that's really an important question, because we really don't know if the Lewy bodies are there and then the disease happens, or maybe they are a survival mechanism that the cell builds up. Maybe they are separate consequences from the disease, and you can hear as many very powerful arguments for each of these theories actually right now.
A question regarding the vector therapies. How do you titrate the dose, and how do you determine the size of the lesion?
So how do you determine the size of the
Number of particles in the lesion.
Right. That's a fantastic question. It's the reason that it's taken a long time until we finally got to the clinical application and to finally start the GDNF trial, which we're about to do, because that was a question that needed to be answered. So there's been a lot of research, a lot of animal experiments looking at this. And in fact, the best we came up with is 4 possible doses. So the study we're doing is actually dose escalation. So we're going to try 4 different dosing regimens to see which one is the appropriate one.
And the outcome should be looking for the increase of the dopamine level?
There are several outcomes that we're looking for. Safety is one, because it's the first application in this form. We are looking for impact on motor symptoms, and then among secondary outcomes, there is a PET, an imaging marker of sorts, looking at the amount of dopamine.
Good luck.
Thank you.
So one other quick question. The implication of some of the therapeutics, particularly the more recent ones, are that they really should be given early in the course of the disease. And so my question is, if it's early in the course of the disease and the symptoms of the very early disease are symptoms which occur with a great variety of other kinds of illnesses, and the diagnosis is based predominantly on clinical symptomatology, you implied that the scanning can be helpful but not diagnostic. How do you resolve that dilemma?
I think you've just outlined a major reason why a lot of research effort is put in, which is the early diagnostic question. I think that's what you're getting at. So we do not have a good early diagnostic solution. There's a lot of work being put into this. Treatments actually, the only reason to give treatment early is if it's a quality of life and to treat symptoms. If we had a neuroprotective treatment, that's where this question becomes major. As of now, we do not have a proven neuroprotective treatment. which is unfortunate. But we all hope that one will come about, and then the question of early diagnosis will become even more acute. So we're hoping to refine ways to make the diagnosis early before we actually have that.
Now, in many mitochondrial diseases, which we'll hear about next week, where the implication is that there's reactive oxygen species and so forth, which is the driving force for much of the pathology, the clinical trials are done with large doses of antioxidants and vitamin E and fish oil and all that, and there is some evidence in some of those diseases of improvement. If that's the common mechanism as proposed in your pathway, is that not a...
Yes, and that's been, so of course research went there. The mitochondrial pathways are definitely involved. However, so far, all the studies, all of that has been tried. Coenzyme Q10, creatine, and vitamin E, and all of that, and one after the other, they have pretty much failed. Some of them still have some hope that maybe there will be some influence on the disease pathway, but nothing has been a game changer. So as of now, as far as we know, nothing has been proven neuroprotective in Parkinson's.
These are mitochondria in neurons, not in
Correct.
other cells.
In neurons, yes.
I think, so we can have time for questions and comments
Thank you very much.
afterwards, so Alexxai would...
Actually, just put on.
Great, can you guys hear me? I want to thank Lynn for inviting me to speak and all of you for coming. What I'm going to talk about is a little bit different. It's going to be mouse work, talking about the some things that can kind of get us thinking of what may be possible in people at some point, or may inform human therapies. And the title of what I'm going to talk is called Optogenetic Control of Basal Ganglia Circuitry. I'm going to have two main points. The first is going to be an optogenetic investigation of this circuitry that was just outlined. And then I'm going to go into a Parkinson's model and talk about how can this work tell us something about Parkinson's disease. And hopefully, by the end, I'm going to leave you with a message that there's two basal ganglia circuits. They've already been brought up, the direct and indirect pathway. They have an opposing control over motor output, kind of as we went through in that diagram. I'll get pretty explicit on this opposing control. And that each one may represent a therapeutic target for treating Parkinson's disease, maybe with some of these kind of new vector therapies or some tricky ways of actually modulating the activity of each. So to talk you through this circuitry, this is the basal ganglia. Again, it's a very simplified model, but the striatum, also referred to in primates as the caudate and putamen. In mice, we lump it together. It seems to be one structure called the striatum. It's the input nucleus of the basal ganglia. It receives inputs from cortical and thalamic structures, and there's two main output groups. These are the direct and indirect pathways that we mentioned before. I'm going to refer to these as the D1 pathway and the D2 pathway, which refers to a dopamine receptor that's expressed differentially on each one. So if we focus on this D1 pathway, or direct pathway, this has our inhibitory cells. They have an inhibitory control over substantia nigra, an area called the reticulata, and these are also inhibitory cells that inhibit motor output. So if you kind of think through this double negative, you come to the idea that if these D1 cells fire, it'll release motor output from inhibition from the nigra, and in this way the D1 pathway can facilitate movement. If we bring back the D2 pathway, here we have the same release from inhibition phenomenon, but on the subthalamic nucleus, which will now excite the nigra. So in this way, when D2 cells fire, the nigra is excited, and the D2 pathway inhibits movement. One more puzzle piece I'm going to add are dopamine neurons themselves. So we talked about the substantia nigra pars compacta, that black area in the brains that we saw. And those are the dopamine neurons themselves. They live in the nigra. They project back to the striatum. And because these two populations of striatal neurons have different dopamine receptors, these dopamine can have a different effect on each one. The D1 receptor is excitatory, so dopamine can excite the D1 receptor and facilitate movement this way. The D2 receptor is inhibitory, so dopamine can inhibit the inhibitory pathway and facilitate movement in that way too. So two independent pathways. The effect of dopamine on both of them is to facilitate movement. For the rest of this talk, I'm going to give you an even more simplified model. This is kind of the two things I want you to remember going through are that there's two pathways, D1 and D2 pathway, or direct and indirect pathway, that have an opposing control of motor output. And then dopamine itself can have a different effect on each pathway because these pathways, the cells that start these pathways have different dopamine receptors. So when I started investigating these about 4 years ago, this was a model, and there was a lot of evidence in support of it, but it had not been empirically tested in a direct way. And the reason that it was that the technology didn't exist. That unfortunately, unlike the schematic I showed where all the D1 cells are on one side and the D2 cells on the other, in real life, these cells are mixed up in the striatum. So this is a slice of a mouse that has a red fluorescent protein in D1 cells, a green fluorescent protein in D2 cells, and a slice of the striatum. You see that they're all mixed in together, and this is true throughout the striatum. So if you take a technique like electrical stimulation, and you stimulate in the striatum, and many people have done this, you're equally likely to stimulate one pathway or both pathways together. So teasing apart the independent function of each pathway at the level of the striatum is not possible with techniques like this. So I became excited about an emerging technique at the time called optogenetics. And this refers to the genetic expression of light-activated proteins in the nervous system, is where it's been most widely applied, but it's also been applied outside of the nervous system. And the main protein I'm going to talk about, this light-activated protein, is channel rhodopsin 2. So this is a light-activated ion channel. It sits on the membrane of a neuron. And it's activated by blue light, and it'll open like an AMPA receptor, depolarize the cell, and cause it to spike. So here's an example of that in a schematic. This is a patch recording of a striatal cell, and every time a blue light pulse happens, you see the cell fires a spike. I'm going to show you a video of this. These are kind of 3 different traces at different frequencies. I'm going to show you a video in an awake mouse. Let me see. Not sure how loud this is going to be. We're going to be listening to little tick sounds. Are going to be a single D1 pathway neuron from the striatum firing. This mouse has a recording array of electrodes into the striatum and a fiber optic that we're seeing illuminated here, that when the light turns on with the fiber optic, it's going to activate the channel rhodopsin in these D1 cells. And we're going to listen to the increase in firing. So you probably get the idea. So we have this in D1 cells. We can selectively activate them in awake mice. And what this allows, one of the really strong benefits of optogenetics over other techniques like electrical stimulation is this cell type specific control. So we're in a structure that contains direct pathway and indirect pathway cells all mixed in together. Instead of where electrical stimulation would activate them both, with optogenetic stimulation, we come in with a fiber optic, and we can selectively activate one or the other depending on which one we've targeted the channel rhodopsin to. For completeness, I'm going to just mention that there's also, we can change the protein itself. So instead of channel rhodopsin, we can use some other proteins that have inhibitory effects on the cell. And then you can do cell type specific optical inhibition as well. And with a preparation like this, we I'm going to take you through the genetic targeting on how we get it into the cell types of interest. And we use a system that relies on Cree mice. A lot of them are generated by a project combined with NIH and Rockefeller University. And this is a mouse that expresses a protein known as Cree just in cells in these D2 cells. We're looking at a coronal section of a mouse brain here. The striatum is this big area in the mouse that's lit up in brown. That's staining for Cree. And if we could see a little bit more closely, we would see that only half the cells are actually stained for Cree, those that express the D2 receptor. This is the D2 Cree mouse. And then we can come in with 2 cannulas and infuse a Cree-dependent channel rhodopsin virus. So this is a virus that will infect all the cells, but it'll only become active in those that express Cre. We did this obviously in D2 Cre and in D1 Cre mice to target the two pathways. And I'm going to show you a little bit of histology. This is work that was done. I did this with two very talented graduate students, Ben Fries and Phil Parker. This was in California. And we're looking at now is a sagittal section of a mouse brain, so looking at it from the side. And we're seeing the three major, the striatum and the two main targets. The pallidus right next door is the main target of the D2 pathway. And then all the way across the brain, it's almost a centimeter, sends these fibers, striatal D1 cells, D1 neurons send fibers to synapse in the nigra. So when we look at histology, now we're looking at the same view, sagittal view, and this is the striatum up here. And the fibers come all the way down here into the nigra. This is a channelrhodopsin in the D1 Cre mouse. If we put the same virus into the D2 Cre mouse, we get, again, the cell bodies in the striatum, and the fibers terminate right next door in the pallidus right there. So now we can do an experiment and test how does stimulation of these pathways affect movement. I'm going to show you a video. In this video, we're going to illuminate the D2 cells. They express channelrhodopsin. And the hypothesis is this is going to inhibit movement, which in fact you'll see it does. In this video, the word laser is going to appear whenever the laser turns on, stimulating these D2 cells. And the effect will be pretty obvious. So this is rapidly reversible. The mouse recovers pretty much immediately. But when these cells are stimulated, this mouse is frozen. You're going to see this 2 more times. So this last pulse is about 15 seconds. I've done these out 4 or 5 minutes for various behavioral reasons, and they really stay frozen. You can kind of think back to the videos that we saw in the previous talk of a freezing of gait, and possibly it may be, kind of make a conjecture that these Parkinsonian patients may be experiencing the freezing of gait because their indirect pathway is stuck on. So I quantified this in a few ways. This is a summary. What we're looking at now are 3 bars. Pre refers to 30 seconds before the laser pulse. Laser is a 30-second laser pulse, and then post is right after 30 seconds. So we see the before the laser, and then the recovery. And in this graph, I'm just splitting up the total time into 3 categories. Freezing is like we saw in the video when he's really frozen, and it's a measure that I get from the pixel changes in the video, so it's really the video frame is not changing. Walking is when the mouse is moving from place to place. And then mice do a lot of other things, that they're staying in place, they're grooming, they're sniffing, things that are a little harder to tease apart. So I'm lumping them together and calling them fine movement here. And as you saw in the video, freezing went way up. It was about 5-fold more freezing in these animals when the laser's on, and it recovers as soon as the laser's off. Walking goes almost to 0, that these mice, when the laser's on, they really don't go anywhere. And then fine movement also dropped a lot simply because they're frozen so much time. Show you the same plot in the D1 channelrhodopsin mice, and now we have the opposite effect, where freezing is pretty much at 0. This laser is not that. That when the D1's down, they're really not frozen. They spend a lot of time walking around, running around the arena, and then their fine movement is also reduced because they're spending so much time running around. I broke this up a little bit finer, and in these plots, now we're looking at every time they move, they're tracked by video. So we can pull out things like how many times did they move, when they do move, how long was the movement, when they do move, how fast is the movement. And that's what I'm showing you here in these 3. The one on the left is how many times did they move, and we're still in that same, let me see if I have a pointer. All right, I don't think I have a... And that's what I'm trying to do, but I'm not having luck with that. Okay, there's this one. So here we're looking again at before the laser, during the laser, after. And we can see for the D2 stimulation, the indirect pathway stim, they move less frequently. When they move, the movements are shorter, and the movements are slower, and like that bradykinesia that we saw in the Parkinson's patients. If we look at the D1 Cre, we get the opposite effect, that they move more frequently. They initiate them. When they move, these movements are longer, and actually they're of the same speed. There were no differences in the speed that they're moving with the D1 stimulation. We can also look at the freezing itself. So the mouse spends a lot of time frozen with the D2 stimulation, but we can ask questions like, is he freezing more often, or does he freeze longer? That's these first two. So he's freezing a little bit more often, but the big effect was that when he freezes, he just doesn't start again. He stays frozen. And then we also looked at fine movement. I've mentioned this is kind of an aggregate of a lot of different behavior, but we can still look at the center point of the mouse and see how fast it's moving. And the fine movement is also slower. Again, that's kind of reminiscent of the bradykinesia in Parkinson's patients. When we bring back we look at the D1 stimulation, they freeze less frequently. And this is kind of the most obvious thing by eye, that when they freeze, a normal mouse kind of walks and freezes and stops here and there. When the laser is on with the D1 mice, they just go immediately. They're just constantly, every time they stop, they start again. So the duration that they're frozen is much reduced. And then also we found with the D1 that their grooming and sniffing and things was more vigorous, and evidence that they have a faster fine movement speed in contrast to the locomotion speed. The final thing that, before we get into the Parkinson's models, was we also looked at gait. We saw that Parkinson's patients, that they have this very stereotyped gait, and we tried to analyze this on a treadmill that was equipped with a high-speed camera that images the mice from below. So we can see all the paw prints. Software can pull out the paws, and it can give you metrics like how long are his strides, how wide is the stance, what are the angles of the paws, is there an increase in variance from step to step with the laser on or off. And at the time, surprisingly, this was a big negative result, that when the laser's on, if they walk, these mice walk normally on the treadmill. And I think at the time I was kind of surprised, but speaking more with clinicians, probably something I should have done earlier, Parkinson's patients also can walk on a treadmill, and the gait problems may be a lot more pronounced in natural walking. So keep in mind, we didn't have any gait differences with the laser on or off. I'm sorry, these bars are representing the percent change from laser on to off. So 100% is no change with the D1 on or the D2 on, and nothing was significant there. But this is a kind of a forced motion that maybe if we had a more voluntary motion where the mouse just walked around and we still imaged him, we may have found differences in gait. So the conclusion for this first part are that the D1 pathway increased both the frequency and the duration of movement. The D2 pathway decreased the frequency and duration of movements. And then we did have some smaller changes in the mechanics of locomotion and the movement speed. The big effects were on the initiation, on when they make movements, how many movements do they make. So I'm now going to move to a Parkinson's model and talk about what this work can tell us about Parkinson's disease. If we go back to the simple model, and even this is the double simple model I made, where really we're looking at the striatum and the effects of the D1 and D2 pathways, positive and negative, on motor output, we can turn this into a Parkinson's model pretty easily. By these cells that make dopamine, they die. So these striatal cells lose their dopamine input. And a prediction based on the excitatory effect of D1 receptors and inhibitory effect of D2 receptors is that we should lose excitatory tone in the D1 pathway and have overactive D2 pathway. And two questions that came from this. One is, would inhibiting the indirect pathway improve Parkinsonian symptoms? And the second is, would activating the direct pathway? I'm not going to talk about the first question. This, in fact, appears to be true, that inhibiting the indirect pathway does improve Parkinsonian symptoms. It's thought to be the main mechanistic effect of deep brain stimulation, that these are kind of turning off indirect pathway structures. But this other idea of could you activate the direct pathway could really only be done in the striatum. This is a monosynaptic connection from the striatum to the nigrus. There's no other place in the circuit that you could activate, just the direct pathway. And it can really only be done in an animal situation like this at this point. that's the question that we focused on, whether we could activate the direct pathway and change some Parkinsonian symptoms. The model that we used was a toxin model. So this is very different than the genetics and the human condition. This is really a model of dopamine cell death. So we're looking at the chemical structure here of dopamine, and then a toxin called 6-hydroxydopamine, and the 6-hydroxy refers to a hydroxyl group on the 6' carbon. This is, because it looks like dopamine, it's taken up by dopamine cells, and it'll end up causing oxidative stress and killing those cells. So keep in mind that it's a model of dopamine cell death, but it's probably relevant sort of to the dopaminergic cell death that's seen in the end-stage Parkinson's. So to show you what this does to the striatum, this is a control brain stain for a protein, an enzyme tyrosine hydroxylase. This is an enzyme in the synthetic pathway of dopamine, so dopamine cells express this enzyme. And if we stain for it, we see the striatum here lit up strongly in red. That's where most of the dopamine terminals are in the brain. We then injected 6-hydroxydopamine through a cannula. You can actually see the little bit of damage from the cannula in this section. And we injected it directly into the striatum. So where the toxin went, those dopamine terminals took up the toxin and they die. And the reason that we did it into the striatum, as opposed to something more global that would affect dopamine throughout the brain, was that our optogenetic manipulations rely on a fiber optic, and the light really doesn't go very far. So in this way, we could go back through the same cannula. We could express chanorodopsin. This is in D1 cells. We're going to test activating D1 cells in a Parkinson's model. And we can express it through the same cannula in the same region. And early on, we thought, if we take dopamine out of the entire striatum with a much bigger lesion, and we only have control of a small piece of tissue, we may not get any beneficial effects. Here we're controlling the same piece of tissue that's missing the dopamine and presumably has hypoactive D1 expressing neurons. So this is the condition, the simplified model of Parkinson's disease with these hypoactive D1 cells. And the test we want to know is, can we turn on the laser, stimulate these cells, and restore motor output? I'm going to show you a video. Again, you'll see the word laser. And in this video, you're going to see the mouse walking around. And if you kind of think back to the other video, or become a parent, this lesion that we did in the striatum is not very robust. It's sort of a moderate Parkinson's lesion. It's only removing dopamine in that area of the striatum. So this mouse moves around okay. He's a little bit slow. One notable thing is these mice don't rear. They kind of do these slow behaviors on the bottom. And the laser is going to turn on now. And he becomes pretty much an exploratory kind of, excited mouse. This laser will stay on for a little longer, and then we'll see it turn off and his behavior return to the Parkinsonian condition. So the laser turned off, and he's rapidly back. So I'm going to show you the same quantification that we did with the control data. Here we're looking at 2 bars, and this first bar is really the effect of the lesion. So I tested every mouse before the lesion to get an idea of their baseline freezing, walking, and fine movement. What we're looking at here is the pre-laser period, but it's the effect of the lesion. That they freeze more, they walk less, fine movement is not significantly different. But these mice, it's mild. They can eat, they can drink, they maintain their weight. When we turned on the laser now, we're seeing the laser and the period where the laser is following the laser. It restored the walking and the freezing behaviors pretty much to the pre-lesion levels, which I found a little surprising. I was somewhat worried about getting too much movement, or some of the things we talked about, the dyskinesias that happen with too much Parkinson's medication or dopaminergic medication. And really, we don't see that. I tried cranking the laser higher and things, and it seems like you can drive these cells as much as possible, and I have not seen dyskinetic behaviors come out. This becomes even more impressive when we break the behavior down into the frequency of initiations, the duration of each movement, and the speed of each movement. And what we saw was that the lesion itself really affects the frequency of initiations. So this is now the mice in pre-lesion and everything normalized to that. So the effect of the lesion was to reduce the frequency, not the duration or the speed. So when these mice would move, they tend to do normal seeming movements based on these two parameters. And when the laser's on, these frequency came right back to previous, not an overshoot. When we look at freezing and fine movement, this was sort of the only overshoot that we had was in freezing. Both the frequency and the duration of freezing was higher in the Parkinsonian animals. And when the laser's on, they freeze less frequently, but the duration is, again, kind of restored right to where it was. And then the same thing with the fine movements, that these mice were a little bit slower with the fine movements, again, like a bradykinesia. And this was restored to that same level. So I'm going to add to this findings that I feel like I skipped this slide earlier. But the main findings of both the first part of the talk were that the direct pathway activation increases movement, the indirect pathway decreased it. I'm going to add to it that this direct pathway activation is therapeutic in this mouse model of Parkinson's disease. And I'm going to end with one final thought of what does this mean for patients? Is this something that we're going to see at some point? And I think about 2 years ago, I actually had this slide, and my point was that this is kind of a better research tool, and there's so many technical hurdles to using this in people that first you need the viral delivery of a protein, you need to get the optogenetic stimulation into people, and I really thought this is a research tool. What you can use it for is learning something about the system and then use some traditional ways to find other ways to stimulate the D1 cells, maybe pharmacological or just identifying it as a target. I think in the last year or so, I've felt less secure in the idea that this is not going to happen in people. I've met people that are designing devices and have patents for putting them in people. You hear talks like this with the gene therapies really coming along. So I don't know, it seems there's no inherent reason why something like this couldn't be applied in people. I'd like to acknowledge the lab where this data was collected. This was during my postdoc with Anatol Kreitzer, and I mentioned a few people that contributed directly to the data that I've shown. And I'll end with the conclusion that I hopefully have convinced you that there's two basal ganglia circuits that have an opposing control over motor output, and each may represent a therapeutic target for treating Parkinson's disease. I'd be happy to take any questions, and certainly if you think of any at a later date, this is my email address. Feel free to drop me a line. Thank you.
Thank you very much. Very exciting.
Thank you. Fascinating. This is fantastic work. First, I just want to say that I'm usually a pessimist, but I remember reading your 2010 paper, and I didn't know you were pessimistic then, because when I read it then, I thought, wow, this should go towards therapy at some point. So I'm curious if you have combined this with electrophysiology. I thought that was where at some point your lecture was going, because I'd be very curious to know what happens to the beta band and all those things when you turn these on and off.
Yeah, so the question is whether this has been combined with electrophysiology and to record from these cells. And I showed an example, this mouse, that clearly we can identify the cells, and then you can kind of track them through a Parkinsonian manipulation. And we have that actually in submission, and I can talk to you about that later. I don't know if I'm going to get into details of that. But.
Are there any neurons that are both dopaminergic and glutaminergic, the same neuron in a sequence?
Yeah, so
how does that conceivably influence what you're...
So we mentioned that this model is extremely simplified, and if we keep asking questions, we'll learn all the ways it's simplified, but one of the simplifications is these neurons actually release glutamate as well, or at least a subset of them does release glutamate. Subset of them may also release GABA. So it's it's trickier than just them releasing dopamine and exciting the direct pathway and inhibiting. I don't know that anyone knows how tricky it gets, but they do know that those cells release glutamate.
I have a question about your scoring for the mouse behavior. Is it subjective or objective? Because you can tell when the laser's on if you're just looking at the mouse. So how do you avoid bias in the observer?
We use computers to observe it. So this is all video tracked, and the computer does know when the laser's on, because that's the way of removing bias.
What are you working on here at the NIH?
So I'm actually taking the same system and researching obesity and food addiction. So one of the surprising things, my background was in addiction, and I learned about this circuitry in the context of addiction. It's the same circuitry, the same dopamine cells that are the effects of addictive drugs are acting on, and the same adaptations in the striatum. So kind of going back to that, but instead of looking at drugs of abuse, looking at different diets and how they may share properties with drugs of abuse in terms of changing the circuit in addictive ways.
Any way to get a double product? I mean, you show that you could both, with optogenetics, you could both inhibit and excite different neurons. Did you manage to do the double product for the D1 and D2s?
To also inhibit them?
At the same time, with the same laser bout?
Not at the same time. Let me see if I have... Yeah, I don't have this data here. So I've never... So I'll answer your question in two ways. One is that you can put both an inhibitory and an excitatory construct into the same virus, and theoretically people have shown the proof of concept that you can go up and down with the firing rate of a cell. In practice, that's been, can be dirtier because now you have two proteins in the membrane. So I've done it separately, using the same system, but putting an inhibitory protein just into these cells. Actually, I haven't done it in the D1. And they move more if you inhibit them. The effects are not nearly as robust as exciting them. So exciting them, you saw this video where the mouse just freezes. When you inhibit them, they move about 20% more. So you really need to do the video analysis and tease out that they're moving more.
Basically, the idea I was going for is that imitating exactly the Parkinsonian model.
Put one in each?
Exactly.
I haven't done that. I think, yeah, there's no...
Each, and then the crossover, which would be the effective treatment eventually.
Yeah, so I haven't done that. There's no technical limitations why that couldn't be done.
For sure.
No, I just was helping you.
We ask that you speak to the microphone because there are many, many people who are online and they can't hear the question.
You had mentioned that you modulated the intensity of the laser. Did you find there was a threshold effect in the motions? That is this a system which is proportional control, or is there a nonlinear threshold in the responses?
It's pretty linear. So the test I did with that was actually a rotation test, where if you stimulate one side, the animal rotates, which is it's kind of easy to quantify. And that went up linear with the intensity of the laser. So getting down to pretty low levels, getting down to about a 20th of what we saw in that.
Thank you.
This is too much. In this video. So that's 1 milliwatt. For reference, laser pointers are usually about 1 to 3. So that's 1 milliwatt getting delivered, and I've gone down to 0.05 milliwatts. So really faint, and you can still get some bias in rotations, and it just goes up with the intensity of the light.
It's continuous modulation.
Yeah.
So what is the depth of penetration you could reach to deliver the laser? What are the laser wavelengths you have tried, and what is the maximum you could reach with the infrared laser?
So the question is, what is the depth of the penetration? And it depends. The depth with this protein, which responds to blue light, we have to use the blue light to activate it, and it's about 1.5 millimeters. With an infrared laser, you could penetrate further, but you'd need to modify the channel rhodopsin to respond to infrared. And people have been doing that, kind of have shown some success, but not success in terms of modulating behavior.
Does blue light penetrate through the skull?
Not very well. Blue light is, in some ways, the worst. It scatters the most.
That's the only one.
So,
Now, the reason I ask is because years ago we helped to develop a technique for treating children with prolonged unconjugated hyperbilirubinemia. This is the Billy lights, which even today newborn kids with marked jaundice are put under the lights. But some of these kids who have a genetic defect where they have lifelong hyperbilirubinemia, they're under the blue lights for more than 12 hours a day for years. And they often have neurological findings which are ascribed to something called kernicterus, which is a toxic manifestation of bilirubin in the same area of the brain, the basal ganglia. And I wonder, in the light of what you're telling us, whether some of those manifestations may not have been the result of the prolonged phototherapy. And this is of great practical importance because these kids wind up having liver transplants to restore the enzymatic defect. Is that possible?
I want to say it's not possible that blue light gets through that way. It may be, it could be retinal. So, and there's special melanopsins that are responsive to blue light that are not visual cells in our retina.
These are newborn infants. Many of them are preemies. Well, we can talk about it. Okay, well, if there are no other questions, I want to thank you both for really a very exciting presentations, and thank you all for being here.